From Kansas State University, this is Agriculture Today. Eric Atkinson with you. Ahead today, a USDA plant pathologist stationed here at K-State, John Fellers, will talk about remarkable advances in DNA sequencing technology that now allow for more rapid identification of virus diseases in wheat. He says that wheat breeding programs will greatly benefit from this. We'll also hear today from K-State's Alison Crane as she'll preview a special workshop taking place this Saturday here at the university. It'll train adults on starting and conducting local 4-H and FFA wool judging teams and activities for youth. Also, this week's horticulture segment, K-State's Dennis Patton on caring for poinsettia plants throughout this Christmas season. All that here on Agriculture Today. Glad to have you along once more for this Agriculture Today. Well, wheat producers know the importance of research into dealing with virus diseases that we see frequently in that leading crop here in the state of Kansas. And so we take this time to bring you up to the moment on some really interesting advances in contending with those viruses, learning more about them now. We've invited over John Fellers. John is a USDA plant pathologist who works with wheat diseases regularly, and he is based here at Kansas State University and certainly interacts with the plant pathology department here at K-State on these ventures. And John, you have some new technology you want to tell us about, but before we do that, virus diseases are a perpetual problem in wheat production in this region, aren't they?
Yes, they are, and especially in the last couple of years have we seen out in western Kansas. We've been seeing new viruses, high wheat streak mosaic complex symptoms. We even have varieties that we've released, especially out of the Hayes program, that have had wheat streak mosaic resistance genes in them, and we thought that those would hold up. But there's questions about it now where you have varieties like Clara CL or Ron L that are supposed to have resistance, but the farmers are coming in and say, hey, it looks like I've got some sort of symptoms on it. What's going on? And so it's been very important. And so we have some research plots in Lane County this year, and they really got hammered. And so it opened up an opportunity for us to really test out some new technology that we have in the lab.
And we want to get into that. But are we seeing something unusual happening in the way of viral adaptability to that resistance? What's going on?
No, I don't think so. I mean, in the world of viruses, we're seeing it, just in our everyday news. We have the Omicron variant of the COVID virus. It's an RNA virus like our wheat one. It's a different family. But viruses, they're known for mutating. They adapt very quickly, and the mechanisms they have to replicate themselves are not very accurate. So when it replicates itself, it makes mutations in there. And so we see when we release virus-resistant varieties, whether it's corn or wheat or soybeans, we sometimes see that in the viruses, they tend to overcome them very quickly. And it's just that their mechanism they have to adapt. And Kansas farmers, well, not just Kansas farmers, but any farmer, if they see their neighbor growing a new variety or they see a new variety, they put it out there. And what happens is we get a lot of the same resistance gene out in the fields. And so it puts a very heavy selection pressure on the pathogens. We see it in leaf rust. Farmers have been dealing with it with leaf rust all the time. Put a new resistance gene out there, 4 or 5 years, that resistance gene doesn't work. And so it's great job security if you're a wheat breeder, but it's not really good if you're a wheat farmer. So even seeing it on my own wheat that I grow in Oklahoma and my family farm. And so, but viruses adapt just as much. It's just that we haven't had tools available to us until the last 5 or 6 years in the form of resistance genes that we've been able to release. I mean, we've had tolerance, but as far as resistance per se, like we see in the rust, we just really haven't had it. And so we've turned some varieties loose, but I think we see just how quickly the virus can adapt.
Then let's talk about your research charge with the USDA in adjunct work with K-State as well here, and some of the technological advances that we've seen happen. Give us an idea of what you are doing specifically and how you're getting to it.
So I'm what's called a molecular biologist. So I do DNA sequencing or genomics work. So anyone that has done the 23andMe and those kind of DNA testing where they send off their DNA and they find out how they're related to people. I do that kind of work for wheat, for leaf rust, and for viruses. So I take in the pathogen and rust, I isolate the DNA, I sequence it. When I first got here, the Wheat Commission funded one of the first big DNA sequencers in my lab, and it was the size of a refrigerator, and it cost $350,000, and every 2 hours it would cost me $1,500 to run. Wow. But it was a big advancement, and every 2 hours we would get 70,000 letters of DNA, and at that point I was working on wheat. They were working on corn, they were working on the human genome, the Arabidopsis genome. The Arabidopsis, the rice, and the corn genomes were about $50 million projects to sequence all of the DNA. The human genome was about $500 million. And all of them were because they had these big machines. And then about 2010, it started dropping. So I got funded to do leaf rust. It was $1 million project, and the technology was changing a lot. But we were able to do it for $500,000, sequence leaf rust. And it's 120. At that point, it was 136 million letters of DNA. So it started getting really cheap. Right. Then in about 2014, 2015, a company out of England developed a technology where in the beginning we had to take pieces of DNA, cut it up, put it into a bacteria, and then pick individual colonies of bacteria. And we had robots that would pick those bacteria. And the process alone, there was $2 million worth of equipment in my lab to do all of this. Now, we don't even have to chop it up. We can just take the leaf of the wheat plant, isolate the DNA, ligate on some DNA primers, and then load it onto a sequencer now that's now the size of a harmonica.
Exactly.
Okay. Yeah, and I'm sorry, I know we're on radio, and it's hard to see it, but literally it is the size of a harmonica. And
you're holding it in your hand as
that's
you
right.
talk.
And I play in a bluegrass band, play harmonica. So, yes, it's that size. But this is now, I can buy the kit. The initial machine that you plug into your laptop is $1,000. The little cell that we put into it is $750. But now I can take a leaf of wheat, isolate the DNA, and in 48 hours I can sequence 10 billion letters of DNA for about $2,000 from the initial one. But each run is less than $1,000.
So it's come a long way.
It's come a very long way.
Economically, but functionally too, you say.
Yes. And so if we have a new leaf rust disease that can come out, we bring it into the lab, we grow it up, and within a week I can sequence the genome of leaf rust now for about $3,000. Remember, it was $500,000 in 2011, so 10 years. And so in 48 hours we can have an answer. But specifically what we're able to do, and it's something that we've all been dealing with for the last 2 years, is with viruses and how we're using it for detection.
This is one of the big steps forward with this technology, not only the reduction in cost and the size convenience, but the fact that you say, John, read multiple viruses at once.
That's right. So, well, we've all been talking about COVID, and we all have we've heard about the antibodies, and you can get an antibody test kit. So basically how that works is you take saliva or whatever they have, and you drop it on there, and as it runs up a little test strip, if the virus protein is in there, it picks up these antibodies. And then when it moves to a certain place, if the antibodies are there, it turns purple. You get a band, okay? People have been doing pregnancy tests. It's the same thing. If the pregnancy proteins are there, you get 2 bands. If not, you don't. So you also can do it with plants. You can buy a little test kit. If your rhododendron has a virus or your irises have a virus, you can grind up the sap, put this little test strip in there, and if there's a band there, if it turns purple, then you have a virus and you know your plant is sick. Well, we can do that same thing, but the thing about those tests, it only detects one virus. So farmers have been for a long time sending samples into the K-State Plant Diagnostic Lab, and they use the same type of tests. And so we know that over the years in wheat, that there's wheat streak, there's triticum mosaic virus, and high plains virus, and barley yellow dwarf. Those are the big four. And so that if they look at the symptoms and they kind of get an idea, okay, these are the ones we need to test. But it only tells you that it's either wheat streak or it's triticum mosaic. It doesn't tell you if the virus is changing. It doesn't tell you what isolate's out there. You have to go into the sequence of the virus and find out what it is. So we've been wanting some sort of new technique or technology to be able to say, okay, we've got wheat streak resistant lines. I've got a field full of Clara CL that's supposed to be resistant, but it's got virus in it. What's happening? And so we would traditionally send that sample in to the diagnostic lab, and they would run their tests. And then they would say, well, unfortunately, it came up positive for wheat streak. Well, it's not supposed to. What's going on? So we've got to have some new technology. So this new technology is just a little harmonica-shaped sequencer. This last year, we put to the test.
And we want to hear about that, how well this technology did perform in genetic identification of virus or viruses in those wheat plots out there in west central Kansas this past year. And ultimately, how this knowledge about the genetic makeup of those viruses can lend to wheat variety development to counter those diseases. But we need to take a break, John, and we'll then pick up on all that afterward. Longtime wheat disease pathologist John Fellers with the U.S. Department of Agriculture based here at Kansas State University. We'll return with him shortly. This is the K-State Radio Network and Agriculture Today. This is Agriculture Today, and resuming our visit now with USDA plant pathologist based here at K-State, John Fellers. John is telling us today about this great advance in genetic sequencing technology that he and his colleagues here at K-State are now using to identify and break down the genetic composition of common virus diseases in wheat. And where we left off, John, ahead of the break, you were getting ready to tell us how well this technology has performed in analyzing viruses. This in wheat test plots in west central Kansas this past growing season. Please fill us in on that.
You bet. We have some research plots in Lane County, and we have the new disease resistance genes to wheat streak out there and some experimental lines. And some of those had symptoms. Some of the lines did not have symptoms. So we went through and we isolated them. And with Jessica Rupp, who is a K-State plant pathologist, and her student, Nar Ranabat, who's from Nepal, we took all these samples, we brought them in, and we sequenced them. We sequenced the RNA. Viruses can either be DNA viruses or RNA viruses. The COVID virus that we're dealing with is an RNA virus. Wheat streak, triticum mosaic, high plains, and barley yellow dwarf, they're RNA viruses too. So we have to turn them into DNA, and then we can put them on the sequencer. And what we found out was that many of these wheat plants don't have just wheat streak in them. They can have barley yellow dwarf. They can also have high plains, and we also have triticum mosaic, which we already knew that they get co-infections, but we didn't know to the extent. And so what we've been able to find out by sequencing the whole virus, not just a small piece of it, is we can go in now and say the virus has changed. So the Clara CL, the RON-Ls that had a gene in it called WSM2, the virus has adapted. And we can see exactly in that virus now where it has adapted.
So this new knowledge then will allow for adjustments in, say, wheat breeding programs more rapidly. That's the point?
That's the point. And it also tells us, too, as the wheat breeder, as Alan Fritz goes into his greenhouse or out in his field, and he sees these lines that look like they're resistant, are they really? And what we found out was some of the lines that look like they're resistant, you can actually go in and they still have virus in them. So it's brought a whole new dimension to how we look at viruses in wheat breeding. We've also, in some of NAR's work, he's been doing sampling across the state over the last 3 years. We're starting to see certain parts of Kansas where a virus we didn't think was there called brome mosaic virus.
Talk about that.
Yeah.
That's a new discovery, isn't it?
It's not so much a new discovery. They've seen it in Ohio, in some of the wheat fields out there. A colleague of mine, Dr. Stewart, she has been doing surveys similar to Ohio in the soft wheat areas, and they've seen brome mosaic virus in some of theirs. We just really haven't seen it here in Kansas yet. And so, it brings a whole new thinking of viruses and how we attack it, and should we be looking for resistance for these other viruses too. And the thing is they all have similar symptoms. And I think that's been the frustrating part for me as a wheat plant pathologist is for the technology that we had, and it's still very useful. Don't get me wrong. I'm not trying to say that the diagnostic labs are no longer useful. They still tell us what's there. And the technology they have are still very useful, and it gives us an initial indication. We still need them. But what we're able to do now is we're actually able to be a little bit more accurate and actually have a better perspective of what really is there and understanding really what's going on out there in the environment of our wheat community here in Kansas. In that, I think this will give us an idea if there's a new virus that comes in. And really, that's really how triticum mosaic virus we discovered it, was that the wheat breeder had a wheat streak mosaic line. It was a progenitor to Ron-L. It had symptoms like wheat streak, and we isolated the RNA out of it, and turned out that's how we found out TrimV was there. But what we're able to do here is just to get in one sequencing run, we can get a survey now of what's going on. Now we can go in and look at Kansas and see what isolate of wheat streak is there. Is it showing up where we're seeing WSM2 lines like Clara, where they're predominantly grown? Are we seeing something else coming in? So I think it's going to give us a really new tool that we're going to be able to do it. And not only that, it's designed so that you can take it into your hotel room.
Exactly. It's portable.
It's very portable. Right. We can just isolate the DNA, add some little adapters on it, and it's about a 15-minute prep once you get the DNA, and then you run it on there. And so that's the power that we have. So what we were doing was, with this technology, was that the breeders brought the samples in from Lane County one night. The next morning, we started isolating DNA or RNA. And then the next day we loaded it, and within 4 days we knew what was going on out there, or at least on the virus side.
Remarkable capacity, this technology. It's opened up new opportunities in diagnosis and identification, in potential breeding solutions against those diseases as they tend to evolve rapidly as well. So who would have imagined that we'd be at this point?
No, we're at the whole goal was the $1,000 genome. And we can do it now. Like I say, a starter kit is $1,500. You just need a laptop. All the software is downloadable. It's open source. And so, I think it's going to open up for a lot of applications on genomic science, not just in agriculture, but I think it'll be able to, an FFA group could go out, if they're doing horticulture, they could look at viruses going on in their greenhouse. I think it would be a way of doing that, to really look at them. So I think it's going to be a lot of fun. I'm having a blast with it. I think it's really going to change the way how we look at virus resistance in crops.
Thanks for reporting on this, John. It's impressive, and it's reaping dividends for Kansas wheat production as we look at that disease complex out there and ways of identifying it, contending with it. Appreciate your time, as always.
You bet.
John Feller's with us, USDA plant pathologist here at K-State, and he interacts regularly and works directly with the plant pathology department here at Kansas State University. And talking about the great advances in technology for sequencing diseases, viruses in this particular instance, that tend to plague our winter wheat production in this region. Well, changing gears here to top off this segment, we've word from the K-State Animal Sciences and Industry Department on happenings this weekend in the area of youth livestock. We'll hand it on over now to Agriculture Today's Shelby Varner and her guest. Shelby?
Coming up this weekend is a youth wool judging coaches clinic being hosted here at Kansas State University. Today, K-State Extension Sheep and Meat Goat Specialist Alison Crane. Who is this workshop for?
The coaching clinic, is really focused on starting a youth wool judging program in the state. So there's a few other states in the country that have a pretty active youth wool judging program, and it serves really well for 4-H and FFA kids to get started in any type of judging. We've seen a lot of increased interest throughout the state of Kansas on the youth side of wool judging. So this is just to help get adults started in how to actually teach wool judging, because it's something that not a lot of people know much about. And so the process of teaching wool knowledge, but especially wool judging, is kind of almost a lost art. So it's really for the adults that are wanting to lead an FFA or 4-H team. So whether that's county agents or leaders within the counties for 4-H, or like I said, the FFA programs at schools. And we've also opened it up to not just Kansas, but Missouri, Oklahoma, Nebraska, any of the surrounding states that have people that might be interested in this. And so it's going to be hosted by myself, but also Dr. Whit Stewart at University of Wyoming as well. And some of our assistant coaches on both those teams are going to be helping us too.
There's also mentioning of you guys are distributing totes. What's in those totes for those people from Kansas?
These are only going to be distributed in Kansas. Maybe later on in the next few years, we'll be able to distribute them or possibly sell them to other programs if they're interested in having it. But these boxes or totes are going to have wool samples that could be used in the implementation of a wool judging program for youth. So it'll have samples ranging from finer wool samples all the way through to our coarser samples that students might run into in these contests, but also have a packet of information of what a contest layout looks like. Also a packet of how to teach the wool judging information.
So what does registration look like for this event?
Registration is online. It's as simple as just Googling K-State sheep and goat. And if you go to the sheep and goat page, it's in the right-hand column of the page, and it'll say the Cowboy Classic Wool Judging Invitational, and you click on that, and then you can either register for the contest, but you want to pick the other option, that's the coaching clinic. And it's just $10 per program, so whether it's a county or a high school, you just register for that county. So even if you're bringing multiple people, it's still just $10. So I'm trying to make it affordable for people to come.
That was K-State Extension Sheep and Meat Goat Specialist Alison Crane sharing about the clinics that is happening this Saturday, December 4th. If you are interested, simply search for K-State Sheep and Goat and look for that registration for the event. For Agriculture Today, I'm Shelby Varner. This is the K-State Radio Network.
Well, for our horticulture segment this week on Agriculture Today, it's about holiday time houseplant care. And for this, we turn to Johnson County Extension Horticulture Agent Dennis Patton for his input here. Dennis, it's only natural that we begin with what presumably is the most popular ornamental plant for the Christmas season, that is, the poinsettia. It's a staple in so many homes this time of the year.
Well, you know, the poinsettia is the traditional Christmas plant. It's the number one selling potted plant in the United States for the holidays. Breeders have done a wonderful job making sure those plants are more durable in the home when they're really abused. So the tricks on poinsettias is protect them from a cold freeze or weather. So if you're out shopping, do it on a warmer day, or protect them. Make sure it's the last thing you do on your shopping trip, because the cold breeze will cause them to drop their foliage. On the other hand, don't put them in a really hot heater vent area too, because that'll cause them to drop their foliage. What tends to happen with improper care, watering, the green leaves fall off, but the colorful, what we call the flower, which is really the bracts, that are red and white and pink and all sorts of colors, those will stay on. So the plant really likes even moisture, doesn't like to be wet, doesn't like to be overly dry, doesn't like the hot or cold drafts. As far as indoors, it doesn't matter whether they're in the sunlight, in more the interior of the room, because a lot of people just consider them a holiday decorating idea. And come January 1st or so, they kind of go out with a lot of the other Christmas decorations.
So they're more of an easy keeper, actually, once you get them indoors and protect them. They are a tropical plant, to remind, so the cold is really the biggest threat to these, isn't it?
Yeah, correct. If you take really good care of it in the home, treat it like a houseplant, you can have it probably in still colorful bracts, probably Easter time before they really start going off. And some people like to keep their poinsettias, and they'll put them outside during the summer months. They'll grow this nice big green bush. It takes some effort to get them into bloom because they require the short days. And so if you have natural light in your house and a sunny window, they'll bloom naturally. But if you have overhead lights on, street lights, then you've got to treat them with short days, long nights, probably starting somewhere in September. So they start showing color around Thanksgiving. Fortunately for us, for consumers, they're not all that expensive of a plant. And the other thing that's amazing is the array of colors. Right. If you're around a large retail garden center that sells poinsettias this time of year, go in there and just look. I mean, there's like 10 shades of red, and then now you got all the pinks, the whites, the yellows, the marbles to kind of spice up the holiday decor.
They're a great ornamental flower, obviously, for this time of the year. Poinsettias, and more broadly, Dennis, plants that have been moved indoors for the winter now, and assuring that they remain in good stead, you have to watch a few things.
Yeah, if you've had your house plants outside during the summer months, you bring them in now, they're going to go through what we call acclimation. That means readjusting to the new conditions, which is usually lower light levels. So sometimes you'll see some leaf drop during the winter months because of low light, which is to be expected. You don't really want to keep your house plants growing vigorously during the winter months with the lower light. A lot of times you just want them to kind of sustain. And so we don't recommend fertilizing during the winter months. Just kind of let them go. Water. Most houseplants are probably better a little bit on the drier side than the too wet side. And then the other thing you just might want to check for are the insect issues. A lot of times when those plants are outside, aphids, mealybugs, other things can happen. And then come around Christmas, after the first of the year, they start really just multiplying generation after generation. You may start seeing them on the stems. You may notice sticky sap on the floor of the plants. And then some insecticidal soaps, things like that. Rubbing alcohol, if they're large insects, really work great to wipe them off, those type of things. With houseplants that have been outside, the goal is just keep them alive. The goal is not necessarily get lots of flowers, robust growth, and then you can boot them back outside next spring for another season.
All right, Dennis, we always appreciate your time and your input, and the best to you during these holidays. Many thanks to you,
Eric.
Dennis Patton with us there, Johnson County Extension Horticulture Agent. Again, our time is away. Thanks for being along with us. Eric Atkinson for Agriculture Today over the K-State Radio Network.